In this discussion, we'll be taking a look at cardiac muscle and also the cardiac conduction system. I want to start out here by saying a couple of words about cardiac muscle in general. First up, you kind of have to think back to your A&P1 days and keep in mind some principles that you probably heard about there, and definitely some principles as it relates to skeletal muscle, because cardiac and skeletal muscle, while obviously being different, are going to share some similarities. First up, usually in A&P1, they talk to you about the fact that cardiac muscle obviously is located in the walls of the heart. It also is going to be striated, so it has that pattern of alternating light and dark bands that you saw associated with skeletal muscle as well. If we take a look at the next image here, alright, you'll notice that within cardiac muscle, you'll see that the cardiac muscle fibers or cells are arranged into repeating units, sarcomeres, same as you saw in skeletal muscle. And you're going to have those alternating thick and thin filaments that produce the characteristic banding pattern that you see within sarcomeres. You'll have the I band, which consists primarily of the thin filament attached to the Z disc or Z line. And then you'll have the area where you have the entire length of the thick filament being, once again, the A band. And remember that there is some overlap between the thick and also the thin filament that produces this kind of darker area, darker shading within the A band. But in the center of that A band, where you lose the overlap between the thick and thin filament, you would have the H zone, which consists only of the thick filament by itself, along with the anchoring M line, which is seen in the middle. So the point that I'm trying to make here is, from a histological perspective, cardiac muscle is similarly arranged, at least, to skeletal muscle. It has the regular repeating units of the sarcomeres arranged to the same banding pattern that you've discussed before. But there are some differences between skeletal and cardiac muscle, obviously, and one of the other differences besides the location is going to be the shape of the cardiac muscle fibers or cells. The shape is a branching pattern. These cells are branched as compared to the skeletal muscle, which would be cylindrical in shape. They're also somewhat, instead of being round, they're somewhat more squarish than the skeletal muscle cells as well. And they typically tend to be a little bit shorter also. Now, thinking back, or looking rather here, at cardiac muscle, you'll usually also see an abundance of mitochondria. Usually you'll see more mitochondria associated with the cardiac muscle due to the fact that it needs to continuously produce ATP. Now, as far as its functioning, cardiac muscle is going to function similar to skeletal muscle. If you remember, in skeletal muscle, you had the action potential generated by the motor neuron. Well, once that action potential was generated, it caused release of calcium from the sarcoplasmic reticulum. And you can see that cardiac muscle also has a sarcoplasmic reticulum. That calcium bound to the troponin tropomyosin complex caused the complex to shift or move. And once that shift occurred, it exposed the myosin binding sites on the thin filament, which permitted the thick filament to pull on the thin filament. And it's a similar pattern that you see here in cardiac muscle. Action potentials are generated. Those action potentials cause release of calcium from the sarcoplasmic reticulum. Once that release of calcium occurs, the troponin tropomyosin complex associated with the thin filament in cardiac muscle will shift. And once that shift occurs, the thick filament is capable of pulling on the thin filament, and therefore you will ultimately see contraction of cardiac muscle as a result of that interaction. Now, when we look at how the heart goes about generating an action potential, a major difference between the heart and skeletal muscle is going to be where the signal for the action potential comes from. And when we look at skeletal muscle, that signal came from a motor neuron, your nervous system basically. But when we look at the heart, the signal for the heart to contract the action potential is going to come from the heart itself. So the heart has a property that we call autorhythmicity, or we say the heart is autorhythmic. It means that the heart sets its own pace of beating. It causes its own contraction. There's a series of components located throughout the heart that have the ability to spontaneously depolarize, or we say sometimes they are self-excitable. We refer to these components as a cardiac conduction system, and it is this cardiac conduction system that will cause the heart to contract. Taking a look here on the left-hand side of the diagram, these are the 5 components of the cardiac conduction system, and they are listed in the order in which the signal for depolarization, or the action potential, should travel through them. You absolutely need to know the proper order that the signal will travel through these components for the purposes of your quiz and test. So you also need to know where these components are located, so we're going to go through that in this discussion here. Let's deal with the first component, which is called the sinoatrial node, or SA node for short. It's located high in the wall of the right atrium. The sinoatrial node is considered the pacemaker of your heart. It is where the signal for your heart to beat or for your heart to contract will originate. What happens is, cells within the SA node have the ability to spontaneously depolarize. Once they depolarize, that wave of depolarization begins to spread over both atria, as you see indicated by the arrows here in the diagram. As that wave spreads, both atria would then depolarize, and consequently, those atria would also contract. The signal continues to spread inferiorly down toward the apex of the heart, but as it does so, it moves through the inner atrial septum, which you see me tracing out right here. Within the inner atrial septum is where we find the second component of the cardiac conduction system, which is the atrioventricular, or just AV node. This AV node is located once again within the walls of the inner atrial septum and is known as the gatekeeper, because the way that the cardiac conduction system sets up is that while one set of chambers, like the atria, are depolarizing, the other set of chambers, the ventricles, should be in a state of relaxation, or they should be what we call repolarized. Well, in order to prevent the state of the chambers from becoming out of sync, in order to prevent a disruption of the synchronization, well, you only allow the signal from the SA node to have one way to travel into the ventricles, and that one way is by traveling through the AV node. Since the AV node is the key for the signal to reach the ventricles, since it's the only way for the signal to reach the ventricles under normal circumstances, the AV node is sometimes nicknamed the gatekeeper. It guards the movement of the depolarization from the atria and into ventricles, and therefore it is critical, or its functioning is critical, in maintaining the synchrony that we see associated with the contraction of the heart. So we have the AV node, once again, the gatekeeper, located within the interatrial septum. Once that signal passes through the AV node, it will then enter the ventricles. The first component of the cardiac conduction system within the walls of the ventricles is called the bundle of His, or the AV bundle, or the atrioventricular bundle. All three are acceptable. The AV bundle is located within what is called the membranous interventricular septum. If you notice around the bundle of His here, there is no muscle. There's just a very thin membranous wall. It's still part of the interventricular septum, so technically speaking at this point we're within the ventricles, but because there's no muscle, we call it the membranous interventricular septum. The depolarization signal continues through the bundle of His and then eventually reaches an area of the interventricular septum where there is muscle. This is called the muscular interventricular septum. Within the muscular interventricular septum, the bundle of His will split and form two branches. They are simply called right and left bundle branches because they are branches from the bundle of His. So through the muscular interventricular septum, we have left and also right bundle branches. Those bundle branches continue inferiorly toward the apex of the heart, and once they reach the apex of the heart, they will turn and run in the outside or lateral walls of the ventricles. In the outside or lateral walls of the ventricles, these are called Purkinje fibers, and they are the fifth and final component of the cardiac conduction system. So once again, the cardiac conduction system is where the signal for contraction originates. The heart is autorhythmic. The signal relays from the SA node to the AV node to the bundle of His to the left and right bundle branches, and then finally the Purkinje fibers. And as we just talked about, I gave you the locations of those 5 components, and you'll also want to make sure that you keep that in mind as well. Now, although the cardiac conduction system will cause your heart to contract, it does not mean that this system cannot be influenced. As we will see later on in this chapter, the cardiac conduction system is what causes the heart to beat. But your nervous and endocrine system have the ability to influence the way in which this cardiac conduction system will work. So basically, although your nervous system and your endocrine system cannot, on their own at least, make your heart beat, they can influence the way in which it beats. And that's kind of a, saying making something beat versus influencing the way it beats. Two different statements. And like I said, later on in this chapter when we deal with the concept called cardiac output, you'll get a little bit more about that. Alright, the next section in the book is something that I'm going to kind of just mention here very briefly. It's not a major point, at least for the purposes of your quiz or test. But your book says a few words about action potentials and contraction of cardiac muscle fibers, as well as ATP production in cardiac muscle. So I'll say a couple of words about that. One of the things that we usually talk about in association with the action potential generated in cardiac muscle is that it has what we call a plateau phase. This plateau phase produces a prolonged exposure of those myosin binding sites. And because you have this prolonged exposure, it allows for the heart to contract for a little bit longer of a period of time. And this makes some sense because when we look at the heart, it has to literally squeeze and force blood out. That's not an instantaneous process. So whereas we look at action potentials in skeletal muscle and we get depolarization followed by quick phase of repolarization, when we look at an action potential in cardiac muscle, we get depolarization followed by a prolonged plateau phase of depolarization. This allows for a longer period of contraction, and essentially this is the time in which those cardiac muscle fibers are squeezing and forcing blood to move in the direction in which it would need to go. One last word here about ATP production in cardiac muscle. Obviously, muscle uses ATP in order to power that contraction cycle. It'll happen the same in cardiac muscle as it does in skeletal muscle. And most of the ATP used by your cardiac muscle is going to be dependent upon aerobic respiration. So that's why you have a higher number of mitochondria in cardiac muscle than you'll see in skeletal muscle. It's because those mitochondria have to continuously carry out aerobic respiration in order to generate enough ATP to allow cardiac muscle to continue to function. There will be some creatine phosphate, if you recall that from AMP1. There is some creatine phosphate located within cardiac muscle that provides some ATP, although it's not going to be a primary source of ATP. Alright, the next topic we want to start talking about here is something that's related to the cardiac conduction system, and that topic is electrocardiograms. EKGs, ECGs, the abbreviations for that topic. An electrocardiogram basically is a picture of the electrical activity of the heart. Now, because that electrical activity is caused by the cardiac conduction cycle, the cardiac conduction system, essentially what an EKG becomes is a picture of how well the cardiac conduction system is working. The EKG is measured as a function of time versus voltage or millivolts. So you see here, this is a standard EKG tracing. It's a standard EKG tracing that would occur during one complete heartbeat, or one what we call cardiac cycle. You can see, on average, this cardiac cycle or one heartbeat takes about 0.8 seconds. So all of these events that occur during a normal, or electrical changes rather at least, that occur during a normal heartbeat occur in under a second, only 0.8 seconds. You can see that the EKG shows a series of deflections away from baseline, so above 0 millivolts. And these deflections all typically mean something or relate to something. Now, for the purposes of your test, you will need to know several standard components to an EKG, and you'll also need to know what they represent from an electrical standpoint, and by that, you're going to hear terms such as depolarization or repolarization. And you'll also need to know what those terms are, what happens from a physical perspective. And from a physical perspective, I mean, is a chamber contracting or is it relaxing? Now, as we go through this, you're going to see some alternative terminology come up, so I'm just going to define it for you right now. When we look at the heart, we have alternative terms referring to contraction and relaxation. When a chamber or chambers of your heart contract, we refer to that contraction as systole, S-Y-S-T-O-L-E. It's just an alternate name, still means contraction. If a chamber of the heart relaxes, we refer to it as diastole. So you have systole and also diastole. Two terms that you're going to see come up here that you should be familiar with. So first up here at time 0, in order to start a heartbeat, you have to have the SA node fired. So if we look at the next diagram here, that's number 1, at time 0, the SA node would fire. As I told you during the cardiac conduction system discussion, that will cause a wave of depolarization to spread over both atria. As that wave of depolarization spreads, we get an electrical change. The EKG shows an upward deflection, and we call this upward deflection the P wave. This P wave represents atrial depolarization. Now, going back to the next diagram here, as that wave of depolarization spreads over both atria, eventually you come to a point where you have depolarized all of the atrial muscle. If you depolarized all of the atrial muscle, you cannot continue to depolarize it. Everything at that point would be depolarized. So instead of continuing to go up, that P wave comes back down to baseline. And this represents the time when those atria are fully depolarized, and when a chamber of the heart depolarizes, it will contract. So at this point at which we have the P wave returning to baseline, the atria are fully depolarized, and you would then be in what we call atrial systole. The atria are fully contracted. And if you notice here on the diagram, the atria are a little bit more rounder, a little bit fuller here on the left, but then they're smaller here on the right to denote that they are in full systole. Now, when you return to baseline, there's some time that elapses. Go back to the previous diagram here. The reason that we return to baseline and that we have this flat segment is because, once again, the atria are in full systole. They're taking time to squeeze and eject blood into the ventricles. Now, the time period from the beginning of the P wave to the beginning or the end of this flat baseline segment is called a PQ interval. This PQ interval basically represents the time that it takes for the signal for depolarization to spread from the SA node and into the bundle of His. Because what's also occurring here during this baseline segment is that the signal for depolarization is being relayed through the AV node. That relay takes time, and that slows the signal down enough so that the atria have time to eject blood into the ventricles below. Once that signal reaches the bundle of His in the interventricular septum, we see the beginning of the next major component of the EKG called the QRS complex. This QRS complex represents a couple of different electrical events. First up, since we have the signal moving through the bundle of His, the left and right bundle branches, and also the Purkinje fibers, we're going to see ventricular depolarization during the QRS complex. But while these ventricles are depolarizing, the atria are also repolarizing. So the two electrical events occurring during the QRS complex are going to be ventricular depolarization coupled with atrial repolarization. Both occur at the same time. Now, why don't you get a nice individual peak for atrial repolarization? It's honestly all about the amount of muscle. Having more muscle in the walls of the ventricles means that you get a greater electrical signal from those ventricles. This will ultimately cause the ventricular signal to cover up or mask the atrial signal in the QRS complex. Well, after completion of the QRS complex, we then have another short baseline segment. This baseline segment represents the time when the ventricles are fully depolarized. In other words, the ventricles would be in systole. This segment also has a name. This is called the ST segment. The ST segment represents the time when the ventricles are in full systole and they at this point will be ejecting blood from the heart. After those ventricles complete systole, they'll then begin to relax or repolarize. And as they begin to relax or repolarize, we get the next major component of the EKG, which is going to be the T wave. The T wave represents ventricular repolarization. Or, if we looked at it physically, we could say ventricular diastole. Finally, after the T wave, we get another return to baseline here, and during this time, all of the chambers would be relaxed. It's a very brief moment in time, 0.2 seconds, 0.2 of a second, but this is a time when the chambers of the heart are simply filling with blood in preparation to contract again. This time period is variable. It doesn't necessarily have a name, but it is variable because if your heart rate increases, this time frame can be decreased as well. It's one of the main places where your heart will begin to cut time down. So those are the major components of the EKG. Like I said, you definitely want to know the electrical event that's occurring, the depolarization, repolarization stuff. You should also be able to correlate that with the physical event as well. All right. That will conclude our discussion, and in the next discussion we'll go ahead and talk about what you see here on the screen, which is the cardiac cycle.